You know, in biology, I think we have this bad habit of ignoring the packaging.
Oh, definitely.
We get so obsessed with the heart, the lungs, the brain, the expensive machinery inside.
Right. But the wrapper,
Yeah.
we just kind of dismiss it, the saran wrap view of anatomy.
Exactly. And that's the perfect example. Take the lung. It's wrapped in this incredibly thin, translucent layer of tissue called the mesothelium. And if you open a standard medical textbook, the dogma has been that it's just a passive lubricant.
Sure, it's there. So your lungs don't chafe against your ribs when you breathe.
That's it, full stop.
Which, to be fair, is a super important job. Friction is bad.
Yeah.
But if you stop there, you miss the real story. Because a new study just came out, Luna et al., in Developmental Cell, that just completely dismantles that idea.
Okay.
They've basically proven that this wrapper is actually a command center. It's not just protecting the lung, it's actively telling the lung how to build itself.
And maybe what's more surprising is that it's fighting this constant, desperate battle to remember who it is.
Right. It's a full-blown identity crisis, but on a cellular level.
That's the hook for me. But the plot twist, the real kicker, is the gene at the center of this. The paper identifies a gene called MYRF. And when I saw that, I assumed I was reading a neurology paper.
Because you know MYRF is a brain gene.
It's the myelin regulatory factor. I mean, it is the gene. It's famous for wrapping our nerves in insulation so we can think and move. So my first question is just, what on earth is a brain insulation gene doing in the lining of the lung?
That is the curveball. And it turns out evolution is very efficient. It reuses its best tools.
Okay.
So in the brain, MYRF builds the myelin sheath, but in the lung, it acts as what we call a lineage gatekeeper.
Our gatekeeper.
Yes. The researchers found that if you delete this single gene from that thin long wrapper, the cells get confused. They forget they're supposed to be these skin-like protective cells, and they start trying to turn into muscle.
And the consequences of that confusion, they change drastically depending on when the gene fails. This is what I want to structure our deep dive around today, because we aren't just talking about one disease here. We're talking about two completely different nightmares.
Right.
One that kills embryos and one that suffocates adults. And it all stems from this one gene failing at different times.
It's a story all about timing.
So let's start at the very, very beginning, the early break. The researchers managed to delete MYRF right at the dawn of lung development.
Embryonic day 6.5. I mean, this is basically when the lung is just a microscopic bud, barely a cluster of cells.
So super early.
Incredibly early. And to be clear on the precision here, they used a genetic driver, think of it as a time switch called TBX4-TTA. This hits the mesothelium before the lung even really looks like a lung.
And the result?
Catastrophic. 100% of these mice developed congenital diaphragmatic hernia, CDH.
Which is a devastating birth defect, and we see this in human babies.
We do. The diaphragm, that muscle sheet separating your chest from your gut, it just fails to close. There's a hole.
Right.
And because of that hole, the stomach, the liver, the intestines, they all start to migrate up into the chest cavity.
Crowding out the heart and the lungs.
Yeah, and specifically on the left side usually. And what's amazing is the mice in this study mirrored that perfectly. Left-sided herniation, just like in humans.
Now, I want to challenge the standard explanation here, because I've read about CDH. The dogma, the thing you hear in med school, is that this is a mechanical problem.
The bad real estate problem.
Yeah, right. The hole allows the stomach to move up. It physically squashes the developing lung, and because the lung has no room, it just stays tiny.
That has been the prevailing theory for decades. Compression causes hypoplasia underdeveloped lungs.
But this paper
flips that entirely on its head.
Yeah.
And this is a massive shift for the field.
So how did they disprove the compression theory?
They were clever. They looked at the lungs before the hernia actually happened. They went back to embryonic day 13.5. So at this stage, the diaphragm is still forming, but the stomach hasn't moved up yet. There is no compression. But when they measured the lungs, they were already tiny.
So the lung was broken before the stomach ever got involved?
Precisely. The defect is intrinsic to the lung. The blueprint was wrong from the start.
So the saran wrap layer, the mesothelium, it wasn't sending the right signals to the lung underneath it.
That's exactly it. They did bulk RNA sequencing on these tiny lungs, which is basically a census of every active gene.
Yeah.
They found radio silence on the growth channels. Specifically, a gene called FGF9 was way, way down.
FGF9, that's fibroblast growth factor 9. That's the signal, right?
It is the primary shout from the wrapper to the lung tissue saying, proliferate, branch out, build more lung.
And without MYRF, the mesothelium just goes mute.
It goes mute. No FGF9.
Oh.
But it gets worse. They also found a massive drop in RALD2 and RDH10.
Okay, those sound like droids from Star Wars. What do those actually do?
They're the little machines, the enzymes, needed to synthesize retinoic acid.
Yeah.
And retinoic acid is a powerful morphogen.
A morphogen. So it's a chemical that tells embryonic cells where they are and what they're supposed to become.
It's the GPS and the instruction manual, all in one.
So without MYRF, the command center fails to send the grow signal, FGF9, and it fails to send the here is your map signal, the retinoic acid.
It's like a gardener who decides to stop watering and stop fertilizing at the exact same time. It doesn't matter how much space the plants have. If they don't get those chemical instructions, they just stunt.
That is a huge insight for CDH. It implies that even if surgeons fix the hole in the diaphragm perfectly, the baby still has lungs that were chemically starved of development signals.
It suggests the lung is fundamentally biologically underdeveloped, not just physically squashed. That completely changes how we have to think about treatment.
Okay, so that's scenario A. You break the gene early, the signaling fails, the lung doesn't grow.
Right.
But the researchers didn't stop there. They asked the next logical question. What if the lung forms okay at first, but loses MYRF later on?
Scenario B, the identity crisis. For this, they switched to a different genetic driver, WT1 CREAR. This one lets them delete the gene later, at embryonic day 12.5.
So by this point, the initial signaling has already happened. The lungs are a decent size.
Yep, the basic structure is there, no hernias.
So from the outside, the mouse looks healthy.
From the outside, maybe. But if you zoom in on that saran wrap layer under a microscope, it's a horror show.
What are we seeing?
Well, normally, mesothelial cells are what we call squamous, think flat pancakes.
Right, a single, organized, smooth layer.
They're designed to be slippery. But in these mutants, the pancakes were gone. The cells had physically changed shape. They turned into these thick, spindle-shaped things.
Spindle-shaped.
Yeah, and they were piling up on top of each other, forming this thick, disorganized rind around the lung.
So they looked almost like scar tissue.
They look like fibroblasts. And this is the key part, like smooth muscle cells.
Huh.
And this is where the genetic detective work gets really cool. They used single-cell RNA sequencing and lineage tracing to tag these cells and see what they were doing.
Okay.
They found the cells had stopped producing the proteins that say, I am a mesothelium genes, like UpK3B and GPM6A.
They lost their ID cards.
They threw away their ID cards. Yep. And in their place, they picked up new ones. They started pumping out a ton of ACTA2.
ACTA2 is smooth muscle actin. That's the classic marker for muscle cells. So the skin of the lung is trying to become muscle.
It's trying, but it's failing. And this is the nuance that makes this study so important. They didn't fully turn into functional muscle cells.
Ah.
They became these confused hybrid cells. They had some skin markers left, but now they had these muscle markers too. They're just stuck in developmental limbo.
So why does the cell default to muscle? That seems random. Why not bone? Why not fat?
That's the million-dollar question, isn't it?
Yeah.
It implies that smooth muscle is the default setting for these cells if they aren't constantly being reminded to be mesothelium.
So identity isn't a static thing you achieve once.
No, it's an active state you have to constantly maintain.
Let's talk about the mechanism here. How does one gene, MYRF, prevent this slide into muscle? The paper used a technique called CTRNUN to figure this out.
Which I still think is the best name for any molecular biology technique.
It does sound like an action movie, but essentially it's just a high-resolution map, right?
Exactly. It lets us see precisely where the MYRF protein physically touches the DNA strand. And what they found was that MYRF binds directly to the enhancers. You can think of those as the volume knobs for those key mesothelial genes we mentioned.
So it's physically holding the switch in the on position for be a wrapper.
That's a great way to put it.
Mm-hmm.
But the paper says it doesn't work alone. It has partners.
And this is where two very famous characters in biology enter the chat, YAP and TAZ.
Ah, yes, the hippo pathway. I love when these two show up.
So what do they do?
YAP and TAZ are sensors. They tell a cell about its environment, specifically mechanical stress, tension, stretching.
So the researchers suspected MYRF was working with them. How did they prove it?
They did something. Well, something brutal. They created double mutants.
Uh-oh.
They knocked out MYRF and TAZ at the same time. An explosion. I mean, if you lose just one, it's bad. If you lose both, the check on cell identity completely collapses. The cells didn't just drift toward muscle, they sprinted. The study showed a massive explosion of ectopic smooth muscle.
Ectopic meaning muscle where it shouldn't be.
Right. So the mental model is this. Imagine MYRF and YAP-TAZ are two security guards leaning against a door.
Okay.
Behind that door is the Become a Muscle program. As long as they're both there, the door stays shut and the cell stays a flat pancake.
Right.
You remove one guard, the door rattles a bit. You remove both, the door bursts open.
That is a fantastic visual. And it sets up the final and I think the most harrowing part of the study, because we've been talking about embryos. But what happens if you let these mice grow up?
This is where the rubber meets the road for human medicine. The researchers induced the gene loss a bit later so the mice wouldn't die from the hernia, and then they just let them age.
And the result creates a perfect model for a rare human disease.
It does. The adult mice developed a condition where the lung surface became encased in a hard fibrous shell. The researchers actually called it a corset.
A corset, that's a suffocating image.
It's physically accurate. The mutant cells, confused and thinking they were muscle-like fibroblasts, just churned out massive amounts of elastin and Act2.
Wow.
They created a thick, stiff rind around the entire lung.
And they actually measured the stiffness using a special ventilator, a Flexivent machine.
They did, yeah. They basically pumped air into the lungs and measured the resistance. The data showed incredibly high elastance.
So stiffness.
Extreme stiffness.
Yeah.
Imagine trying to inflate a balloon that has been coated in duct tape. That is what these mice were breathing with.
And this specific pattern, this thickening at the surface, especially in the upper lobes, highly elastic. It looks exactly like a human disease called PPFE.
Pleuroparenchymal fibroelastosis.
Which is a mouthful.
It is, and it's a terrible diagnosis. It's rare and until now really poorly understood. In PPFE, patients get this fibrosis in the upper lungs, specifically at the pleura, at the wrapper. Most other fibrosis diseases like IPF start deep in the tissue and march outward. This one starts at the surface and squeezes in.
Because the problem is in the wrapper itself.
Exactly. And the scariest part, the researchers described what they called a zombie effect.
This part gave me chills.
Right. Usually, if a cell gets this confused, it triggers apoptosis. It commits suicide for the greater good of the organism.
Sure.
But these mutant cells, they survive. They just persisted for a year, which is old age for mouse, just sitting there churning out this fibrous shell, creating this permanent corset.
So let's just pull this all together for a second.
Yeah.
We have this graded level of evidence that is really robust. We have the genetic link variants of MYRF have been found in human CDH patients.
Yep.
We have the cellular evidence, the physical shift from a pancake to a spindle. And now we have the functional evidence, the stiff corset lung in adult mice that perfectly mimics PPFE.
It really redefines MYRF. It's not just a myelin gene anymore. It is a lineage constraint factor.
Lineage constraint. I want to double click on that term.
It implies that a cell's identity isn't some passive default state. It's an active choice that requires constant energy and constant signals to maintain.
So the mesothelium could be muscle. It has that potential.
Exactly. And MYRF is the constraint, the gatekeeper, that stops it from remembering that.
Which brings me to the provocative thought I wanted to leave our listeners with.
Okay.
We established that YAP and TAZ, the partners of MYRF, are tension sensors. They respond to mechanical stretching.
Correct. They read the physics of the cell.
And what does a lung do 24 hours a day from the moment you take your first breath until the moment you die?
It stretches. It breathes. It is a constantly moving, dynamic organ.
So here is the question. Is the act of breathing itself part of the signal that keeps everything in check?
That is the hypothesis this paper just blows wide open. Is the rhythmic stretching of the lung part of what keeps MYRF and YAPTs active?
Does the mechanical force of breathing physically keep those security guards at the door?
And if that's true, think about the implications for medical care. If you put a patient on a ventilator that changes the pressure and the stretch in the lungs.
Or if someone has a lung injury that creates scar tissue and changes how the lung stretches.
Does that change the mechanics enough to make the gene slip? Does the wrapper stop being a wrapper because it stopped feeling the breath correctly?
It's a wild thought.
It is. It suggests that physics and genetics are talking to each other constantly. The way you breathe might literally be telling your genes which proteins to make.
It means that taking a breath is technically a genetic instruction.
I think that's a fair way to put it. Biology is rarely static. It's a constant loop.
Well, on that note, I'm going to go take a very conscious, very deep breath.
Highly recommended.
Thank your mesothelium for knowing who it is. Thank your MYRF gene for keeping the door shut on that muscle identity. And thank you all for listening to this deep dive.
See you next time.